In this video, I would like to explain about beta oxidation of fatty acid. Before going to the beta oxidation, we want to know about the general structure of fatty acids. This is the general structure of fatty acid. Here we can see a straight chain of an even number of carbon atoms with hydrogen atoms along the length of the chain and at one end, and a carboxyl group at the other end. Here you can see a carboxyl group, and here you can see CH2 groups. And when we are naming this is the alpha carbon, this is beta, and this is gamma, etc. So this is the beta carbon. Now coming to the beta oxidation. Beta oxidation is defined as the oxidation at the beta carbon atom. Oxidation of fatty acids at the beta carbon atom with the sequential removal of the 2-carbon compound acetyl-CoA. The structure of the acetyl-CoA is CH3, C double bond O, SCOA. This is the structure of the acetyl-CoA. So in the beta oxidation, the 2-carbon compound acetyl-CoA is released. So beta oxidation is defined as the oxidation of the fatty acids at the beta carbon with the sequential removal of the 2-carbon compound acetyl-CoA. The fatty acids in our body are mostly oxidized by the beta oxidation. The beta oxidation of fatty acid involves 3 steps. They are activation of fatty acids which occur in the cytosol. Second one is transport of fatty acid into mitochondria. Then third one is beta oxidation proper in the mitochondrial matrix. These are the 3 stages of the beta oxidation. First one is the activation of fatty acid which occurs in the cytosol. Second one is the transport of the activated fatty acid into mitochondria. And third one is the beta oxidation proper, which takes place in the mitochondrial matrix. In our body, fatty acids are oxidized in most of the tissues. In our body, the fatty acids are oxidized by most of the tissues, but brain, erythrocyte and adrenal medulla, which cannot utilize fatty acids for energy requirement. They depend on glucose for their energy. So brain, erythrocyte and adrenal medulla, they cannot utilize the fatty acid for energy requirement. Now coming to the first step of beta oxidation, that is fatty acid activation. The fatty acids are activated to acyl-CoA. The fatty acids are activated to acyl-CoA by the enzyme thiokinase. Fatty acids are activated to acyl-CoA by the enzyme thiokinase or acyl-CoA synthetase. Thiokinase is also known as acyl-CoA synthetase. This reaction occurs in 2 steps and requires ATP and Mg2+. This reaction occurs in 2 steps and requires ATP and Mg2+. The fatty acids react with ATP to form acyl adenylate. The fatty acid react with ATP to form acyl adenylate, which then combines with coenzyme A to form acyl-CoA, which then combines with the coenzyme A to form acyl-CoA. Here the AMP is released. Here 2 high energy phosphates are used. 2 high energy phosphates are used because ATP is converted to pyrophosphate. ATP is converted to pyrophosphate. The enzyme inorganic pyrophosphatase, which hydrolyzes PPI to inorganic phosphate. And the immediate elimination of the PPI makes this reaction totally irreversible. So the immediate elimination makes this reaction totally irreversible. So in the first step, the fatty acid react with ATP to form acyl adenylate. This acyl adenylate then combines with the coenzyme A to form acyl-CoA. Here the ATP and Mg2+ is utilized. and here the inorganic pyrophosphate is released. This is converted to inorganic phosphate by the enzyme pyrophosphatase. So two high energy phosphates are utilized in this reaction. And thiokinase is the enzyme which catalyzes this reaction. So it first converts fatty acid to acyl adenylate, then to acyl CoA. Now coming to the second step, that is transport of acyl CoA into mitochondria. Transport of acyl CoA into mitochondria. The inner mitochondrial membrane is impermeable to fatty acids because the activation takes place in the cytosol, and the inner mitochondrial membrane is impermeable to the fatty acid. So a specialized carnitine carrier system which operates to transport the activated fatty acid from the cytosol to mitochondria. So the specialized carnitine carrier system which transport the activated fatty acid from the cytosol to the mitochondria, and this occurs in 4 steps. The first step is the acyl group of acyl CoA. The acyl group of acyl CoA is transferred to carnitine. To form acyl carnitine, which is catalyzed by the enzyme carnitine acyl transferase. The acyl group of acyl CoA is transferred to carnitine to form acyl carnitine, which is catalyzed by the enzyme carnitine acyl transferase 1, which is present in the outer surface of the inner mitochondrial membrane. Carnitine, also known as beta hydroxy gamma trimethyl aminobutyrate. Beta hydroxy gamma trimethyl aminobutyrate. In the second step, the acyl carnitine is transported to mitochondrial matrix. The acyl carnitine is transported across the membrane to the mitochondrial matrix by a specific carrier protein. So in the second step, the acyl carnitine is transported to the mitochondrial matrix through the mitochondrial membrane. By a specific carrier protein. Now the acyl carnitine is in the mitochondrial matrix. And inside the matrix, the carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane. Carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane, converts the acyl carnitine to acyl CoA with the release of carnitine and the use of coenzyme A. So the acyl carnitine is converted to acyl CoA with the release of the carnitine and using the coenzyme A. This reaction is catalyzed by the enzyme carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane. In the third step, the carnitine released is returned to the cytosol. For reuse. I repeat once more. The inner mitochondrial membrane is impermeable to fatty acids. So a specialized carnitine carrier system operates to transport the activated fatty acid from the cytosol to the mitochondria. So this occurs in 4 steps. First one is the acyl group of acyl CoA is transferred to carnitine to form acyl carnitine. The acyl group of acyl CoA is transferred to carnitine to form acyl carnitine by the enzyme carnitine acyl transferase 1, which is present in the outer surface of the inner mitochondrial membrane, and here one coenzyme A is released. In the second step, the acyl carnitine is transported to the mitochondrial matrix through the inner mitochondrial membrane with the help of a special carrier protein. So now the acyl carnitine is inside the mitochondrial matrix. In the third step, The carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane, which releases the acyl CoA and carnitine, which forms the acyl CoA and releases the carnitine. So the carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane, convert the acyl carnitine to acyl CoA and carnitine. The released carnitine is transported to the cytosol for reuse. These are the steps in the transport of acyl CoA into mitochondria. Now coming to the third step of beta oxidation, that is beta oxidation proper, which takes place in the mitochondria. Each cycle of beta oxidation liberating a 2-carbon unit acetyl CoA, which occurs in a sequence of 4 reactions. That is, each cycle of beta oxidation liberating the 2-carbon compound acetyl CoA, CH3, C double bond O, S CoA. So this occurs in a sequence of 4 reactions. So the first one is oxidation. In this step, the acyl CoA undergoes dehydrogenation by an FAD dependent flavoenzyme, acyl CoA dehydrogenase. So the acyl CoA undergoes dehydrogenation by an FAD dependent enzyme, acyl CoA dehydrogenase. So a double bond is formed between the alpha and beta carbon. Here you can see the double bond. So the double bond is formed between the alpha and beta carbon. So the compound is delta 2 trans-enoyl CoA. Delta 2 trans-enoyl CoA. In the first step of oxidation, the acyl CoA undergo dehydrogenation by an FAD dependent flavoenzyme acyl CoA dehydrogenase to form delta 2 trans-enoyl CoA. Here a double bond is formed between the alpha and beta carbon. Next step is hydration. In this step, the enoyl CoA hydratase enzyme brings about the hydration in the double bond, to form beta hydroxy acyl CoA. Here one water molecule is added and forming the double bond is broken and forming CHOH CH2. So beta hydroxy acyl CoA is formed by the enzyme enoyl CoA hydratase. The reaction is hydration reaction. Third step is again oxidation, that is the beta hydroxy acyl CoA dehydrogenase catalyzes the second oxidation and generates the NADH. Beta hydroxy acyl CoA is converted to beta keto acyl CoA by the enzyme beta hydroxy acyl CoA dehydrogenase. Beta hydroxy acyl CoA dehydrogenase with the generation of the NADH. Here one NAD is used and converted to NADH. So the product formed is beta keto acyl CoA. And the fourth step is cleavage. The final reaction in the beta oxidation is the cleavage. Here the liberation of a 2-carbon fragment acetyl CoA from the acyl CoA. Liberation of the 2-carbon compound acetyl CoA from the acyl CoA by the enzyme thiolase. So this is a thiolytic cleavage and catalyzed by beta keto acyl CoA thiolase. Here one coenzyme EA is used to form the acetyl CoA. So we get a 2-carbon less acyl CoA and acetyl CoA, one acetyl CoA. So the fourth step is the cleavage, that is the final reaction in which the liberation of the 2-carbon fragment acetyl CoA from acyl CoA, and this occurs by a thiolytic cleavage by the enzyme beta keto acyl CoA thiolase, and forming a 2-carbon less acyl CoA and acetyl CoA. So the new acyl CoA which containing 2 carbon less than the original, which re-enters the beta oxidation cycle, and this process continues till the fatty acid is completely oxidized. So the overall reaction is CN acyl CoA plus FAD plus NAD plus H2O plus coenzyme A gives CN minus 2 acyl CoA plus acetyl CoA plus FADH2 plus NADH plus H plus. So this is the overall reaction of the beta oxidation. Beta oxidation is the oxidation of fatty acids on the beta carbon atom, which is the sequential removal of the acetyl CoA, 2 carbon compound acetyl CoA, and this mainly occurs in 3 steps. First one is the activation of fatty acid, which occurs in the cytosol. Then second one is the transport of fatty acid into mitochondria, and then beta oxidation proper in the mitochondrial matrix. So these are all about beta oxidation.